I'm Jörg Ehrman. I'm a rheumatologist at Brigham and Women's Hospital in Boston and vice chair of Spartan. This is the second in a series of 6 global education seminars that are jointly organized by experts from GRAPPA and Spartan. I'm proud to mention that the first of these seminars in January was attended by participants from 32 countries around the world. The recordings of this seminar are also available on the Spartan and GRAPPA websites for other people to watch. For today's seminar, we have registrations from more than 40 countries. So good morning, good afternoon, and good evening, depending on where you are right now. Today's seminar will focus on aspects of genetics and pathophysiology of psoriasis, psoriatic arthritis, and axial spondyloarthritis. Because of time limits, we had to make some choices with regard to content. So for instance, we decided not to get into the role of the intestinal microbiota in the pathogenesis of psoriatic disease and spondyloarthritis. Please use the post-seminar evaluation to let us know what you thought about the content and whether the volume and degree of detail were appropriate for you as the target audience. Next slide. Oh, we have this already. No, go back. So this is an overview of the agenda. So Niti Goel will start off the seminar. Afterwards, we'll have 3 presentations by Kurt de Vlaam, Louise Puig, and Chris Richland, and each of these presentations will be framed by a couple of multiple choice questions. We are going to record the responses anonymously, and so please participate even if you are not sure 100% what the correct answer is. But your responses will help the organizers of this program to going forward and to adjust the program accordingly. We can monitor the success in our teaching. We'll have a question-answer session at the end of the program. All 4 presenters will be available live, and so please stick around and use the opportunity in the chat to ask questions throughout the presentations. Next slide. This is the last introductory slide. I would like to thank Pfizer for supporting this program. Next slide. So without much further ado, let's get started. The first presentation is by Niti Goel. Dr. Goel is a rheumatologist at Duke University in North Carolina. She is also a patient with psoriatic arthritis, and the title of her presentation is Patient Perspective on Genetics and Pathophysiology.
Hi, I'm Niti Goel. I'm a patient research partner from the United States, and I'm also a rheumatologist. I first began experiencing psoriatic arthritis in December 2009. Although I diagnosed myself with psoriatic arthritis within 6 months after the first onset of acute symptoms, it was at least 3 years before I sought treatment for it formally from another physician. In general, with my psoriatic arthritis, I do not have significant psoriasis. I have had a dark patch on my left elbow, as well as what I call defective 5th toenails ever since I was a child, but these were not recognized as being potentially part of my psoriatic disease until I developed the arthritis. My arthritis symptoms started with a cold that I experienced, and I woke up one morning with lower back pain. Shortly thereafter, I had a massage scheduled with the massage therapist who noted that the area on my back, which hurt, was warm to touch. Then over the next month, I developed bilateral heel pain, then my hands and wrists swelled up, and I had pain on the balls of my feet. I initially thought I had axial spondyloarthropathy, but subsequently I started to get nail changes in both of my big toenails, which was onycholysis. That is what tipped me off that I had psoriatic arthritis. I didn't seek help right away, mainly because I needed to get some personal things sorted out in my life, but I kept symptoms at bay by self-treating with medications that didn't work very well. Subsequently, after I finally got my life issues sorted, I did go to a rheumatologist Once I started treatment that actually worked for my psoriatic arthritis, I realized that a lot of symptoms that I had had since I was a teenager, like locking of my left elbow, pain in my jaw, and neck pain, were probably related to the psoriatic arthritis since they resolved with the treatment. Though I know of no one else in my family with psoriatic arthritis or axial spondyloarthropathy, my children have had some early symptoms of disease. However, it is too soon to diagnose them with anything definite, though one has had dactylitis and jaw pain. I have, however, had physicians give me information related to my children in terms of their genetics that doesn't necessarily apply to them because I am of Asian Indian ethnicity. I would like healthcare providers to be better about discussing the genetic diversity of this condition and to relay information with the appropriate context related to the genetics. I would also like to know my children's risk of disease and if there's anything I could do to prevent the condition worsening or developing at all in them. In addition, in speaking with other patient research partners, things that were important for us to understand related to the pathogenesis and genetics of these diseases were the triggers for flare and increased disease activity, prognosis, how to influence disease outcomes, and predictors of treatment response, as well as the possibility of individualized therapy. When discussing data with us, please make sure to be clear about recognizing if we're a member of a population at risk. If the study included, for example, mostly individuals of European ancestry, acknowledge the findings may not be applicable to those of us of different races and ethnicities, and clarify any assumptions that you've made based on the available data or lack thereof. Thank you so much for allowing me to share my story and perspectives as a patient research partner.
Thank you, Niti. I look forward to meeting you on the virtual stage at the end of this seminar. So let's go to our first set of pre-questions. Next slide. So question number 1, an HLA-B27 positive woman with ankylosing spondylitis comes to the clinic accompanied by her 6-month-old son. Assuming that the son is also HLA-B27, what is his approximate risk for developing ankylosing spondylitis? Is this 0.2 to 1%, 2%, 20%, 50%, or 90% if the father is also HLA-B27 positive? You have 5 seconds to answer this question. 5, 4, 3, 2, 1. Okay, we have a clear favorite here. Let's see whether that is also true for the next question. Next slide. Question number 2. Which of the following statements about the psoriasis risk allele HLA-C0602 is correct? It is absent in indigenous populations in South America, Southern Africa, and Australia. It is a major risk factor for all types of psoriasis. It is associated with a lower risk for HIV and HCV infection. It confers an increased risk for the development of psoriatic arthritis, or it is associated with a better response to treatment with ustekinumab. Again, 5 seconds to answer. 5, 4, 3, 2, 1. All right, we have a wider spread of answers here. And with that, I would like to hand over the podium to Dr. Kurt de Vlaam. He is a professor of rheumatologist in Leuven in Belgium, and he's going to discuss the genetics of axial spondyloarthritis, psoriatic arthritis, and psoriasis.
Welcome to the session about genetics in axial spondyloarthritis, psoriasis, and psoriatic arthritis. We will discuss the role of genetics in the susceptibility of the different diseases in the different clinical phenotypes and other potential applications. I have no disclosures to this presentation. The spondyloarthropathy concept groups a number of different clinical entities with common clinical, immunological, and genetic features. Clinically, you can divide the group into predominantly axial spondyloarthritis and predominantly peripheral spondyloarthritis. Ankylosing spondylitis is the hallmark of the axial group. Psoriatic arthritis is typically representing the peripheral group. Clinical features include spondylitis, sacroiliitis, peripheral arthritis, enthesitis, and dactylitis. Patients can also present extra-articular manifestations such as colitis, psoriasis, and uveitis. Besides ankylosing spondylitis and psoriatic arthritis, IBD-related arthritis, juvenile spondyloarthritis, reactive arthritis, and undifferentiated spondyloarthritis belongs to the concept. They share also genetic features, topic of this presentation, and they have inflammatory pathways in common, resulting in overlapping treating modalities. In the early 70s, HLA studies indicated an increased association between some HLA genes and ankylosing spondylitis and psoriasis. The association of ankylosing spondylitis with HLA-B27 was found almost simultaneously by two different groups, just before HLA-W17 was found to be associated with psoriasis. Studies for ankylosing spondylitis about disease concordance in twins and families, and also later in psoriasis, confirmed that genetic factors largely contribute to the susceptibility of these diseases. This is nicely illustrated by the fourth generation pedigree you see from the Iceland family affected by psoriasis. In the case of ankylosing spondylitis, the hereditability is estimated around 90% in the twin studies and confirmed around 63% in later GWAS studies. For psoriasis and psoriatic arthritis, the hereditability is somewhat lower, but all these findings underscore the importance of the contribution of different genes in these diseases. There is a group of diseases that seems to be associated with a number of MHC class I molecules, the so-called MHC class I-opathies. As in the case of AS, we see HLA-B27. In the case of psoriasis, we found HLA-CW6. In the case of ulcerative colitis, we found HLA-BW4. But also other diseases like Behçet disease are associated with an MHC class I molecule. It is clear that in this group the strongest association is the one between ankylosing spondylitis and HLA-B27. Over 85% of AS patients are positive for HLA-B27. This also highlights the fact that some patients with AS are not positive for HLA-B27. In that case, an association can be found with HLA-B60, now called B40. Recently, also an association with one of the familiar Mediterranean fever genes is reported in a Turkish population. Interestingly, the association with MHC class I molecule suggests also some mechanistic clues. MHC class I molecules, such as HLA-B27, are expressed by all cell types and play a role in antigen presentation. The MHC class I pathies have some common features and are completely different from other autoimmune diseases like rheumatoid arthritis and systemic lupus erythematosus. These autoimmune diseases, they show a female predominance, there is no liquid injury and there is never gut involvement, and they are associated with MHC class II molecules. For joint disease, autoimmune disease have most of the time synovial involvement and a polyarticular distribution, while the diseases belonging to the MHC1-opathies have most of the time involvement at the sites of mechanical stress. This means at the antithesis and in the lower limbs. Some years ago, McGonigal suggested a new classification with a continuum between monogenic autoinflammatory diseases at the one end and monogenic autoimmune diseases at the other end. In between, you find polygenic autoimmune and autoinflammatory diseases, and some diseases show overlap with autoinflammation and autoimmunity, such as ankylosing spondylitis and psoriatic arthritis. The actual hypothesis is that gene variants can cause immune dysregulation after mechanical stress or interaction with environmental factors, which are ubiquitous, such as infection, trauma, and repetitive loading. So, what kind of genes are we looking for? Well, we look for genes that increase susceptibility, genes that are associated with certain clinical features of phenotypes, such as articular disease, extra-articular disease, or even comorbidities, and genes for disease outcome or treatment response. Genetic variance modeling has indicated that genetic factors contribute to over 90% to the overall susceptibility in ankylosing spondylitis. HLA-B27 shows one of the strongest associations of an HLA molecule with the disease and is the most important gene for developing ankylosing spondylitis. Another very important gene seems to be ERAP1, but variations in ERAP1 only contribute to the disease development in the presence of HLA-B27, a phenomenon called epistaxis. Thus, ERAP1 alone do not increase the risk for developing ankylosing spondylitis. ERAP1 as a molecule trims peptide to the optimal length before being loading on the MHC class I molecule. The association with both HLA-27 and ERAP1 shows that the antigen presentation and processing might play an important role in the pathogenesis of spondyloarthritis. Since these both genes do not confer the complete genetic risk, other genes must be involved. Other genes actually definitely associated are IL-23 receptor, a family member of the kinesin family 21B, an IL-1 receptor, anthraxin receptor 2, and STAT3. For other additional genes, confirmations in different populations is still needed. You should be aware that most of the actual results of GWAS studies were performed in patients with an European or Chinese ancestry and are not by definition generalizable to all AS patients from any ethnic origin. So additional studies in other populations are needed. The involvement of IL-23 receptor, kinesin family member 21B, IL-1 receptor, anthraxin receptor 2, and STAT3 implicate that genes related to innate immunity and to the Th17 pathway also play a role in the susceptibility of ankylosing spondylitis. The variation in IL-1 receptor 1 and 2 alleles points towards the innate immune response, and the allele variation in IL-23 receptor and STAT3 show that regulation of the Th17 pathway plays also an important role in the susceptibility. Also, genes in the acquired immune response like RANGS3 play a role in the CD8 T cell function are important. HLA-B27 is not only associated with ankylosing spondylitis but also with the other spondyloarthropathy members, but to a lesser extent. The highest frequency is found in juvenile spondyloarthropathy and undifferentiated spondyloarthritis, and to a lesser extent to the axial forms of IBD-related arthritis, psoriatic arthritis, and reactive arthritis. The association of HLA-B27 with AS is found in almost all ethnic groups around the globe, but the prevalence is varying amongst the different ethnicities. The lowest prevalence and thus the lowest association is found in the sub-Saharan region. This does not mean that AS doesn't exist there or is not present, but it is present to a lower extent and possibly associated with other HLA types. Recently, an association with HLA-B7 and B40 was reported in this region, but still need confirmation. Actually, Over 100 protein coding HLA-B27 variants exist, but not all are associated with ankylosing spondylitis. HLA-B27, 05, 02 and 04 are definitely associated. HLA-B27, 06 and 09 are definitely not. The others, it is still unknown and need further research. The risk for developing ankylosing spondylitis is related to the presence of absence of B27 and is best studied in the Caucasian population. The frequency of HLA-B27 in normal Caucasian individuals ranges from 8 to 40%. HLA-B27 in Caucasian AS patients is present in over 90% of the patients. The prevalence of AS in this population ranges from 0.2 to 1.4%, and the prevalence of ankylosing spondylitis in random B27 Caucasian individuals is 2%. But the prevalence is much higher in B27 positive relatives of AS patients and is almost absent in B27 negative relatives of AS patients. So, already mentioned, absence of HLA-B27 do not exclude the diagnosis of AS, but it is clear that HLA-B27 positive patients differ in many aspects from HLA-B27 negative patients. HLA-B27 positive patients has an earlier age of onset and earlier age at diagnosis. They have a greater familiar occurrence. They are more associated with acute anterior uveitis, but less with psoriasis and IBD. They show a better response to TNF inhibitors and show an association with ERAP1. For psoriasis, we see that in the 80s, HLA-CW6 has been recognized as the PSOR1 gene and confers to the susceptibility of psoriasis development. Nonetheless, CW6 is not associated with all aspects of the skin disease. CW6 is associated with psoriasis, but not with scalp psoriasis, nail psoriasis, or intergluteal psoriasis, the major risk factors for developing psoriatic arthritis. CW6 is part of a haplotype associated with early onset type of psoriasis, type 1 psoriasis. A haplotype is a combination of genes on a unique chromosome which are transmitted together. Additional alleles in this haplotype still must be defined. Again, the association of an HLA class 1 molecule points towards a role for antigen presentation in the susceptibility of psoriasis. Since the identification of CW6, many more genes were found associated with psoriasis and psoriatic arthritis. Actually, more genes associated with psoriasis are found than with psoriatic arthritis. The challenge to find genes associated with psoriatic arthritis is that actually most genes found in psoriatic arthritis are also associated with psoriasis and thus not unique for psoriatic arthritis. Actual findings, mainly based on genome-wide association studies, show that different biological pathways are implicated in the susceptibility to psoriasis and psoriatic arthritis and its pathogenesis. Besides genes playing a role in antigen presentation, the regulation of Th17 pathways and innate immune response, genes implicated in the skin barrier function like LCE3 and corneodesmin, and genes in the interferon type 1 signaling pathway contribute to the susceptibility. At the moment, few genes are exclusively associated with psoriatic arthritis. There are associations with HLA B38, 39 and B27 and also genes in the IL-1 gene complex and with variants of IL-13. One of the problems in PSA is the phenotypic heterogeneity and the case ascertainment. Actually, we have some HLA association with the different clinical phenotypes in psoriatic arthritis. Joint deformity and ankylosis are associated with HLA-B8. Dactylitis is associated with the haplotype B8, B27, and enthesitis is associated with B8 also, and all this compared to patients with cutaneous psoriasis or controls. So why should we go for identifying more genes? Well, genetics has proven to be a powerful tool to improve genetic risk prediction, to find new drug targets, and to identify novel pathways that provide important clues to the pathogenesis and newer treatments. To date, most genetics are studied in respect to the disease susceptibility, but genetics must go beyond susceptibility. Various strategies can be employed to study genetic factors that influence the complete conceptual trajectory from risk of psoriasis through disease onset, prognosis to patient outcomes, and other aspects you see as onset of disease, severity, comorbid burden, and response to treatment. Recently, data became available of a potential genetic association with treatment response. Patients with the CW0602 allele have a better and longer response to ustekinumab compared to the patients who were negative for CW0602. Within this C0602 group, patients having additionally the CO4 genotype has significantly more chance to have a sustained PASI 100 response compared to the other groups. Actually, we are living a genetic revolution with the development of new technologies and new analytical methods leading to more global projects, including studying different populations. In the recent years, the combination of GWAS with meta-analysis has led to the discovery of many new genes, especially genes with a lower allele frequency. I like to conclude stating that genetics in spondyloarthritis, psoriatic arthritis, and psoriasis are rapidly evolving due to the advent of new technologies and methodologies. It is unclear if genetics will improve the diagnostics, but recent findings help to better understand pathophysiology, and recent findings helps also to develop new drugs and to make a more optimal treatment choice for our patients. Thank you.
Thank you. Let's go back to our questions. So question number one, an HLA-B27 positive woman with ankylosing spondylitis comes to the clinic accompanied by her 6-month-old son. Assuming that he's also HLA-B27, what is his approximate risk for developing ankylosing spondylitis? 0.221%, 2%, 20%, 50%, or 90% if the father is also HLA-B27 positive. And you have 5 seconds to answer this. 5, 4, 3, 2, 1. Okay, so we have one clear front runner with a slightly higher percentage, and that is the correct answer, 20%. And I would like to welcome Kurt de Vlaam.
Hello.
Good afternoon.
Good afternoon. Good morning.
Do you want to quickly comment on this question?
Yes, we know all that HLA-B27 is almost a mandatory gene to develop the disease, at least the classical one. But you see that the fact that the risk is higher in offsprings than amongst the populations shows also that, in fact, with the B27, probably there are other genes co-segregating with this HLA-B27 gene, and which makes that, in fact, the risk of offsprings of positive parents increases compared to the normal population.
Now, we've known about this association with spondyloarthritis, or initially ankylosing spondylitis, for almost 50 years. What is your front runner as a hypothesis on how to explain this association?
Well, there are a number of theories available. There's the arthritogenic peptide theory. There is the ER stress model theory. There is also the homodimer theory, and there is recently also the mucosal immunodeficiency theory. And I think the last one is a very interesting one, because with the increasing insights of the role of microbiome in developing of different diseases, I think the last one is really worth to explore further and see if this might, let's say, better explain why we develop the disease. But ultimately, it could be that different mechanisms are present in different patients and different families, because as it is a multigenic disease, it's fairly possible that in one type person of one family, a different set of genes is responsible for the disease than in other families or other people, but always in relation to HLA-B27 or class 1 molecules.
Okay, thanks. Let's go to the other question. So post question number 2, which of the following statements about the psoriasis risk allele HLA-C0602 is correct? It is absent in indigenous populations in South America, Southern Africa, and Australia. It's a major risk factor for all types of psoriasis. It is associated with a lower risk for HIV and HCV infection. It confers an increased risk for the development of psoriatic arthritis, or it is associated with a better response to treatment with ustekinumab. 5 seconds to answer. 5, 4, 3, 2, 1. All right, so 60% answered with the last response, and that is the correct answer. So this allele is associated with a better response. Kurt, what is known about potential mechanisms to explain this association with between HLA allele and treatment with a cytokine inhibiting antibody?
Well, let's say the role of CW06 is not very well known, so it's not yet very clear what the relation is with the true biology, and especially what the role is with production or the biology of interleukin 12 and 23. So I think there's still a lot to do. But interestingly, it's I think within rheumatology, it's one of the quite highest associations with a genetic factor, and a quite hard outcome, because the relation here was with PASI 100, and I think that's a very interesting one. And if we see that the increase of response or the chance to obtain this PASI 100 was quite high, if you have this particular set of alleles, then it's promising that in the future probably also genotyping might help to predict response over time, and maybe in the next step to choose therapy. But for that, we still need to have more knowledge about the relation between this, let's say, for example, here C06 and the biology targeted by this specific therapies.
Okay, one last quick question. You use 2 different types of nomenclature to refer to this allele, HLA-CW6 and then HLA-C0602. Can you quickly explain what the difference is?
Well, it's in the nomenclature. There are a number of variants within the CW6. There are a number of subtypes, and especially with the 0602 allele, there is a correlation, and not with the others.
Okay, thanks. Now let's move on, and we'll have you back at the end of the seminar. Before the next presentation, we have 2 more questions. So next slide. Here. Question number 3. According to current pathogenetic models, what does the initiation of psoriasis have in common with physiological responses to skin injury? There's a strong activation of IL-36 and IL-17C production by keratinocytes. There's an innate response mediated by mast cells and polymorphonuclear cells. There is type 1 interferon production by plasmacytoid dendritic cells, activation of myeloid dendritic cells via TNF, or there's upregulation of autoantigens such as ADAMTSL5 and LL37. 5 seconds to answer. 5, 4, 3, 2, 1. Okay, next question. Which of the following antibody target pairings is incorrect? Bimekizumab and IL-17RA, brodalumab and IL-17C, spesolimab and IL-36 receptor, risankizumab and IL-23p19, or ustekinumab and IL-12p40. 5 seconds to answer. 5, 4, 3, 2, 1. Thank you. So that should be an informative presentation that's going to come now. And with that, I would like to introduce Dr. Luis Puig. He is a professor of dermatology in Barcelona, and he is going to discuss cytokines, disease phenotypes, and drug targets.
Hello, my name is Luis Puig. I'm a member of the scientific committee of GRAPPA, and I'm going to talk about cytokines, disease phenotypes, and drug targets. There are no disclosures for this presentation except the sponsor of this whole educational program. And the topics that will be covered are the cytokine networks in psoriasis, focused on psoriasis vulgaris, the phenotypic and endotypic correlation and therapeutic interventions, with some comment on psoriatic arthritis and spondyloarthropathies. Whenever dermatologists look at typical plaque psoriasis, the histopathological correlation pops up in their minds. Keratinocyte proliferation determines typical psoriasiform hyperplasia of the epidermis. Their altered differentiation determines thickening of the stratum corneum with retention of nuclei. And their immunologic activation releases chemokines and lymphokines that attract polymorphonuclears and lymphomononuclear cells and determine dermal edema, blood vessel dilation and proliferation, and signs of ongoing inflammation. The initiation and maintenance of psoriasis can be considered an unrestrained activation of physiological responses to skin injury, such as the type 1 interferon-dependent response to microbiota and skin injury, which activate toll-like receptors and determine a robust innate immune response with increased expression of antimicrobial peptides, which also occurs in psoriasis. On the other hand, the adaptive arm of immunity is also activated by autoantigens such as ADAMTSL5 or cathelicidin, also known as LL37, and even interleukin-26, which bind DNA and would activate toll-like receptors in plasmacytoid dendritic cells and lead to interferon release and activation of myeloid dendritic cells, which would present those antigens both in the lymph nodes and in situ to naive T cells. This early phase of psoriasis vulgaris would be predominant in acute and stable or erythrodermic variants, rather endotypes, of psoriasis. In post-streptococcal guttate psoriasis, there would be a prominent adaptive response by a restricted repertoire of T cells directed against streptococcal epitopes shared by keratinocytes. On the other hand, stable lesions of plaque psoriasis would be defined by a marked adaptive response with predominant activation and persistent polarization of T17 lymphocytes driven by interleukin-23, which is produced mostly by myeloid dendritic cells. T17 cells release interleukin-17A and interleukin-17F, which synergize with tumor necrosis factor alpha to activate the immunologic response of the keratinocyte, whereas interleukin-22 would drive the epidermal proliferation. Activated keratinocytes would release chemokines and lymphokines that attract several types of immune cells, amplifying the inflammatory response. Mast cells and polymorphonuclear leukocytes would contribute further preform interleukin-17 and release proteases that would activate pro-interleukin-36 produced by keratinocytes in a self-amplified loop. Interleukin-1 and interleukin-36 would be especially relevant as regards expression and maintenance of the pustular endotype characterized by predominantly polymorphonuclear intraepidermal collections. Whereas interleukin-12 and tumor necrosis factor have amplifying roles, interleukin-23 is the main regulatory cytokine in psoriasis and drives differentiation and maintenance of T17 and T22 populations. Interestingly, tissue-resident cells expressing interleukin-23 receptors are responsible for fast relapses of psoriasis in previously involved but apparently cured locations by producing high levels of interleukin-22 and interleukin-17. Interleukin-22 determines the keratinocyte proliferative response in association with interleukin-26 and interleukin-29. Interleukin-17A and F also activate the keratinocyte to amplify the inflammatory response and are also responsible for endothelial changes and other manifestations of inflammation in psoriasis. The role of tumor necrosis factor is synergistic with interleukin-17, partly by increasing the expression of interleukin-17 receptors in the membranes of target cells. Immune-activated keratinocytes release numerous chemokines and lymphokines that attract both innate and adaptive immune cells and potentiate the inflammatory infiltrate. Keratinocyte production of interleukin-36 and interleukin-17C create a self-amplifying loop that promotes an increased expression of antimicrobial peptides and the release of a wide spectrum of pro-inflammatory cytokines that contribute to epidermal proliferation as well as to local and systemic inflammation. Thus, in the last decade, there has been a shift in focus in our understanding of the pathogenesis of psoriasis from T-cell activation driven by antigen presentation with IL-23 as the main regulatory cytokine, towards a focus on innate immunity with a central role of the keratinocyte not only as a target, but also as a producer of cytokines that activate dendritic cells, promote local T17 polarization, and participate in self-amplifying loops. Furthermore, the role of polymorphonuclears and mast cells contributing large amounts of preformed IL-17 and proteases that activate pro-interleukin-36 is highly important. The role of IL-36 as a pathogenic mediator of inflammation and epidermal proliferation in psoriasis was initially recognized in the imiquimod murine model, but found its final confirmation in the identification of monogenic variants of autosomal recessive psoriasis or sporadic cases of psoriasis with some phenotypic variation, but characterized in most cases by the appearance of widespread pustules and systemic symptoms corresponding to autoinflammatory diseases. This disease is due to loss-of-function mutations in the interleukin-36 antagonist gene that determine unrestrained activation of the interleukin-36 receptor. Only 1/3 of sporadic cases harbor these mutations, but those who do have earlier onset and more systemic symptoms. Generalized pustular psoriasis has been known for many years with three main phenotypes: early onset, to which most of the monogenic cases correspond, late onset or von Thunbusch disease, and impetigo herpetiformis in pregnant patients. A recent proposal of classification highlights the systemic component of the disease, its frequent association with psoriasis vulgaris, and the relapsing versus persistent course of the disease. Localized variants also exist, either with palmoplantar or involvement or destruction of the nail apparatus, and can coexist with the generalized form in one given patient or in different members of the same family. The histopathological hallmark of the disease are intraepidermal pustules with variable dermal inflammatory infiltrate and epidermal hyperplasia depending on the evolution of the lesions. Several additional mutations have been recognized in pustular psoriasis variants, and there is some phenotypical variability in members of the same family and even in a given individual. But the net result of these mutations is enhanced activation of nuclear factor kappa B in the keratinocyte, which determines release of interleukin-36. Keratinocytes are the major source of interleukin-36 in the skin, and the expression of interleukin-36 can be induced by other pro-inflammatory cytokines, including interleukin-1, tumor necrosis factor, and interleukin-17A. Interleukin-36 is secreted as full-length pro-interleukin-36 that becomes cleaved by neutrophil-derived proteases into a truncated form that has more than 500-fold greater biological activity than the pro-interleukin-36. This truncated interleukin-36 can act back on keratinocytes via the interleukin-36 receptor to induce even more interleukin-36 expression, as well as expression of neutrophil chemokines. The latest identified mutation inactivates an inhibitor of neutrophil proteases. This cartoon summarizes the central role of the keratinocyte in driving localized and systemic inflammation. Interleukin-17, interleukin-1, and tumor necrosis factor alpha induce release of chemokines that amplify this loop, as well as self-potentiating loops of interleukin-17C, interleukin-17E, tumor necrosis factor alpha, and the cytokines of the interleukin-20 family. Interleukin-36 becomes activated by neutrophil proteases and skin-derived cathepsin and signals on keratinocytes, monocytes, and other cells, leading to release of interleukin-6 and systemic inflammation. Interleukin-36, combined with other keratinocyte-derived cytokines, activates dendritic cells to express interleukin-36 that acts on plasmacytoid dendritic cells, inducing release of interferon alpha and further systemic inflammation. Dendritic cells release interleukin-1 beta, interleukin-6, and interleukin-23, which close this adaptive immune cell loop. The recognition of the central role of interleukin-36 in pustular psoriasis and polymorphonuclear infiltration in psoriasis has put the focus on the cytokines of the interleukin-1 family with their complex activating and inhibitory roles, which seem to be active both in psoriasis and in psoriatic arthritis, as recently reviewed by the Pizzalis group. Therapeutic drugging of this pathway has started to provide very promising results in generalized pustular psoriasis with at least one monoclonal antibody directed against the interleukin-36 receptor, spesolimab, in phase 3 of clinical development. As a matter of fact, the application of translational research to the therapeutic armamentarium in psoriasis has yielded extraordinary results. The first class of molecules were directed against tumor necrosis factor alpha, but their efficacy in psoriasis was at least matched by ustekinumab, directed against the P40 subunit of interleukin-12 and interleukin-23, with an excellent safety record and a very advantageous frequency of administration. The advent of interleukin-17 antagonist pushed PASI 90 response rate beyond 70% at 12 weeks. The efficacy of these drugs in psoriatic arthritis and spondyloarthritis is comparable to that of adalimumab. Secukinumab and ixekizumab must be administered every 4 weeks and require high induction in the first year. Brodalumab blocks the receptor IL-17RA and thus signaling by multiple isoforms of IL-17. Bimekizumab, a dual inhibitor of interleukin-17A and interleukin-17F, is in advanced stage of clinical development. Another dual inhibitor nanobody, sonelokimab, is also under development. The latest class of specific P19 or interleukin-23 inhibitors is generally pushing the complete clearance rate beyond 50% to 60% at 16 weeks. They are administered every 8 to 12 weeks with good maintenance of response over time and persistence of the therapeutic effect beyond pharmacokinetic expectations, that is, remission, potential disease modification, and a safety record that matches that of ustekinumab. This network meta-analysis illustrates the short-term and long-term efficacy of the available drugs for the treatment of psoriasis. Of note, the very low placebo response rate at PASI 90 or PASI 100 levels of clearance. The best performers, ixekizumab and brodalumab among the IL-17 inhibitors, and guselkumab and risankizumab among the P19 inhibitors. Risankizumab seems to lead the pack over long term, but the clinical relevance of this evidence in real life remains to be seen. Thus, the interleukin-23, interleukin-17 pathway has proven to be of central therapeutic importance in psoriasis. Interleukin-17 antagonists have the fastest onset of action, especially brodalumab, which inhibits the interleukin-17 receptor A and the signaling of multiple interleukin-17 isoforms, followed by ixekizumab. Guselkumab and risankizumab are P19 inhibitors characterized by high levels of clearance, which is maintained over long term and decreases slowly upon discontinuation. The median decay of PASI 90 response is 24 and 36 weeks since the last dose, respectively. From the safety perspective, TNF-alpha blockade may decrease antitumor immunity and depletes granulomatous response against intracellular pathogens such as Mycobacterium tuberculosis. IL-17 inhibition is associated with increased candida infections and some risk of inflammatory bowel disease exacerbations, whereas interleukin-23 inhibition is devoid of these concerns, probably because of redundance of the immune response. Interestingly enough, newly developed oral agents blocking the relative subunit of tyrosine kinase 2 and thus intracellular signaling by IL-23 and other cytokines might provide an alternative option with similar efficacy to ustekinumab and an acceptable safety profile in clinical trials. This table summarizes the differences between classes of biological agents targeting cytokines in psoriatic disease. Of note, drug immunogenicity decreases drug survival in anti-TNF-alpha agents and contributes to the fact that other classes be the preferred choice by dermatologists. Psoriatic arthritis and spondyloarthritis share with psoriasis many pathogenic cytokines, but the target cells are mesenchymal stem cells and osteoclast precursors, and the final activity inducing bone erosion or bone formation depends on modulation by interleukin-1, interleukin-6, and tumor necrosis factor, as well as rank and its ligand. On the other hand, mechanical trauma is the main trigger of the inflammatory response in the enthesis and the chondro-osseous interface. Many cell types producing T17 cytokines are potentially independent of regulation by interleukin-23, which would explain the differences in response to interleukin-23 and interleukin-17 inhibitors, especially on axial disease. I would like to wrap up with some take-home messages. Both innate and adaptive immune responses are involved in the pathogenesis of psoriatic disease and spondyloarthritis. Interleukin-17 is the main effector cytokine contributing to both inflammation and structural changes. It acts in synergy with tumor necrosis factor and is mainly regulated by interleukin-23. Activated keratinocytes in psoriasis release multiple chemokines and cytokines that amplify inflammation and epidermal hyperplasia. Interleukin-36 in particular is quintessential in driving pustular psoriasis. Translational research has led to the development of several classes of biologicals with unprecedented efficacy in psoriasis. Cytokine networks in psoriatic arthritis and spondyloarthritis are similar to those involved in the inflammation in psoriasis, but anatomical differences and specific cell types involved in bone remodulation contribute to differences in the efficacy of available treatments. Finally, I want to finish with a view of my hospital in Barcelona City, which I hope you can visit soon. Thank you very much.
Thank you very much. I think you'll have a lot of visitors once the COVID-19 pandemic is over. Let's go to the post-test. Question number 3. According to current pathogenetic models, what does the initiation of psoriasis have in common with physiological responses to skin injury? Strong activation of IL-36 and IL-17C production by keratinocytes, innate response mediated by mast cells and polymorphonuclear cells, a type 1 interferon production by plasmacytoid dendritic cells, the activation of myeloid dendritic cells via TNF, or the upregulation of autoantigens such as ADAM-TSL5 and LL37. 5 seconds to answer. 5, 4, 3, 2, 1. Okay. And two favored responses, the third and the fifth. And the correct answer is, next slide, type 1 interferon production by plasma cytodendritic cells. Luis, do you want to comment on the correct answer?
Yes, just to comment that it's a very recent publication, the one that has shown that there are microbiome-derived DNA that activate also some other innate immune activation that engages toll-like receptors in plasmacytoid dendritic cells, and these lead to an increased production of interferon alpha. And this is what eventually turns up all the inflammation and the immune activation, both in psoriasis and in the response against injury. And the proliferation of keratinocytes in injury is in healing of erosions and other lesions is mediated also by interleukin 17 and interleukin 22 in humans. The upregulation of autoantigens is mostly driven by the adaptive arm of immunity in psoriasis. And the activation of interleukin 36 and interleukin 17C is released. They are released by keratinocytes. They are activated. Interleukin 36 is activated by polymononuclear cells, but they are mostly relevant in terms of inflammation, neutrophil-rich inflammation, not in the response against common injuries.
There was a related question in the chat, and it's about subphenotypes of keratinocytes. So recent studies in the joints in rheumatoid arthritis and psoriatic arthritis with single-cell techniques have shown that there are subsets of fibroblasts in the inflamed joints. Is there anything known about similar subsets of keratinocytes?
No, these studies have not been done, to my knowledge, in skin. Most of the studies on the regulation and activation of keratinocytes are based on confluent cultures of normal keratinocytes, not those from psoriasis, and also there are plenty of studies on the imiquimod murine model, but I'm not aware of these single-cell studies in psoriasis as regards interleukin 36.
Yeah, okay. Let's go to the next question. Post question number 4. Which of the following antibody target pairings is incorrect? Bimekizumab and IL-17RA, brodalumab and IL-17C, spesolimab and IL-36 receptor, risankizumab and IL-23p19, or ustekinumab and IL-12p40. 5 seconds to answer. 5, 4, 3, 2, 1. Okay. So 2 front runners, and I think that needs some explanations. So the correct answer, if we go to the next slide, is A.
Yes, this is the main goal of this question was to make sure everybody understood that bimekizumab is a blocker of both, is a dual blocking antibody that blocks interleukin 17F, the F subunit, and the A subunit. So it blocks interleukin 17AA, AF, and FF. This is in contrast to brodalumab. Brodalumab is an antagonist of the interleukin 17 receptor A. It's a subunit which is common to interleukin 17 A, F, C, 25, etc. So this is important because interleukin 17C, which seems to be produced by keratinocytes, is also blocked by brodalumab through its action on the receptor. And this was what I think it was important to remember.
You pointed out that IL-36 is part of the IL-1 family of cytokines.
Yes.
And there was a question in the chat here about the role of IL-1 itself in psoriatic disease. Can you comment on that?
IL-1 and mostly IL-1 beta is important in inducing the systemic inflammation in many responses, and its blockade by anakinra or canakinumab has been attempted and with some success in treating generalized pustular psoriasis. But this is rather let's say bookshot approach. It is the blockade of IL-36 receptor is much more specific, because eventually whenever the inflammasome gets activated in any kind of inflammation, interleukin-1 eventually goes out the immune cells.
One last question from the chat here, and that's about the what is known about the differential response to IL-23 and IL-12 inhibition in psoriatic disease.
That's a very interesting issue. It seems that the interleukin-12, at least in some murine models, might have an anti-inflammatory reaction. And also when we are blocking P19, we are just blocking the different cytokines which are made of P19 as one of their subunits. And all of these cytokines are pro-inflammatory. Whereas when we block P40, it seems that some of the cytokines, even monomers, can be anti-inflammatory. So this might provide some explanation why one drug which was initially targeted towards interleukin-12 eventually was shown to be active through interleukin-23. But when we are using drugs which are specific for the P19 subunit of interleukin-23, we get much better results than with ustekinumab.
Obviously, because it's just one drug that's available, we don't really know whether it's just that one drug or whether this is really...
Briakinumab seemed to be more potent than ustekinumab in the initial trials, but eventually there was some safety issue, some safety signal.
Okay, thanks. We'll have you back on stage at the end of the seminar. Let's go to the next pre-question. Number 5, which of the following statements about tissue-resident memory T cells is correct? These are long-lived cells that continuously circulate through non-lymphoid tissues. They have a limited T-cell receptor repertoire. They are lymphoid cells, but do not have a T-cell receptor. They express CD69 and CD103. Or they provide a very rapid antigen non-specific cytokine response to local injury or infection. 5, 4, 3, 2, 1. Okay. Next slide. Question number 6. Which statement describes best the role of IL-22 in psoriatic arthritis? IL-22 provides key proliferative signals to dermal fibroblasts in the skin, is the major mediator of pathological new bone formation in the spine. It is a critical activator of osteoclasts in peripheral joints. It mediates the recruitment of regulatory T cells to sites of inflammation, or it may play a limited role outside of the skin. 5 seconds to answer. 5, 4, 3, 2, 1. Okay. So the man who is going to answer all these questions is Chris Richlin, whom I welcome to the virtual podium. Dr. Richlin is a professor of rheumatology at the University of Rochester in New York, and he is going to talk about navigating the immune landscape in psoriasis and spondyloarthritis.
Hello, I'm Christopher Richlin, and I'd like to talk to you about navigating the immune landscape in psoriasis and spondyloarthritis. I have no disclosures for this presentation. I'd like to talk about psoriasis and SPA pathogenesis, focusing on interactions of distinct cell populations through activation of multiple pathways. I'd like to discuss these interactions and pathways emphasizing tissue complexity and cellular plasticity, tissue resident memory CD8 T cells or TRM cells, talk about the cytokine effects on tissue epithelial and stromal populations, and then close on immune and resonant cell crosstalk in entheses and bone. This slide illustrates the tissue complexity that we encounter in psoriatic disease. I'd like to make 3 points from this slide. Number one is that a number of tissues are involved in psoriatic arthritis, ranging from the skin, the bone marrow, the gut, the entheses, and the joints. Number two, there are a number of parallel pathways that are operative in these different tissues, which include, of course, TNF pathways as well as the IL-23, IL-17 pathway. And there are various phenotypes that one can see in the different tissues. I suspect that there are also different pathways of activation in the different tissues, even though there's overlapping of cytokines. For example, in the skin, we have a dominant phenotype of keratinocyte biology, whereas in the joint, we're talking more about resident fibroblasts, macrophages, and osteoclasts, as well as cartilage and chondrocytes. So there are contrasts in not only the immune cells and the resident cells, but also likely in the types of cytokine pathways that are being activated. So so-called endotypes, which are driving phenotypes. There's also great plasticity within the cellular compartment. And here we see a Th17 cell. And unlike Th1 and Th2 cells, which have very specific transcriptional programs that are highly regulated, for example, a Th1 cell with gamma interferon, the major cytokine, and Tbet. In the case of Th17, there are a variety of different transcriptional factors that can be activated that can lead to the release of a whole array of cytokines, which can have signals that drive pathogenic events, inhibitory events for pathogenesis, and then non-pathogenic signals. And you can see here the different transcription factors. One can see here the variety of cytokines that can be released. And so just because a cell expresses CCR6 and other markers of being Th17, that does not necessarily tell you what this cell is doing. In order to understand that, one needs to look at single-cell transcriptomics. So this is a really important point, and that's why the complexity both at the tissue level and the cellular level is really quite high and requires special techniques to understand what's going on in individual tissues in individual patients. So here's an example of how monocyte macrophages can take on different roles within the synovial tissue. In a beautiful work published by Kuhlmann and others, they show that in the synovial lining, there are macrophages shown here in red, and these macrophages are forming a barrier, a protective barrier for the synovial lining cells, the fibroblast-like synoviocytes in blue shown here. And you can see in animal models of arthritis that this barrier is disrupted, and now these cells are moving out into various inflammatory tissues or pannus. Here is a very beautiful electron micrograph showing these monocytes, which actually have barrier-like features you might see in epithelial cells, and adjacent to them are fibroblasts and connective tissue in the subsynovial area and also in the other part of the lining. It's well known from work that they showed that these cells derive from CX3CR1 monocytes, and that they can replenish those monocytes that are present as barrier cells in the lining coming from the subsynovial tissue. Well, CX3CR1 cells are not only in the barrier in the joint, but in this study they showed that this population is a source of tissue-specific osteoclasts. So focusing on the synovial lining in this mouse model and performing single-cell RNA-seq, they were able to isolate this P1 population that had features of osteoclasts. And in additional work I don't have time to go into, they were able to show that this osteoclast, like those border monocytes, derive from CX3CR1 monocytes which come to the joint, and depending on the signals that they receive, specific chemokine signals, or in the case of the osteoclast precursor, MCSF, followed by rank ligand, they can develop into tissue-specific osteoclasts that are specific to the arthritis tissue and are different from those osteoclasts we see in other sites of the body. So here again is this complexity and plasticity which comes up at the tissue level as well in the cellular differentiation pathways. So let's talk briefly about tissue-resident memory cells. These are cells that obviously are T cells, and they migrate to a tissue and they stay in that tissue. They're resident, they do not leave. They express CD69 and a subset of them express CD103. They provide rapid on-site response and reactivation. Their highest number at sites of primary activation, so sites where you might have had some type of cut or inoculation or insult in the lung or in the GI tract. And also the joint, as you'll soon see. They're long-lived, but they do not recirculate, and they're antigen-specific. They have alpha-beta receptors that are specific in the skin, the gut, and the lung for a variety of pathogens as shown here. These are the cell surface markers you can see. I'm focusing on the CD8, IL-17 positive cells, which as you see will be important in psoriasis and PSA. They express CD69, as I mentioned, which is important for inhibiting them from leaving their tissue. It blocks the CYP1 receptor. The T cell receptor, which is an alpha-beta receptor. In this case, the cells we're going to be looking at are CD8. They also express CD103 and interestingly PD1, which in other T cells can be a marker of exhaustion along with granzyme B, which these cells can express. But these markers are not ones of exhaustion in this cell subpopulation, but ones of activation. These cells are extremely activated and can be pathogenic. And this slide illustrates that fact so that they're known to have important roles in protecting against pathogens, long-lived responses, whereas they can also participate in allergy, injury, and autoantigen exposure, and they have been shown to be critically important effectors in psoriasis, and I'm about to show you psoriatic arthritis, and many other diseases. So this study showed that IL-17 positive CD8 cells seem to be preferentially found in the synovial fluid rather than the peripheral blood of patients with PSA. They found similarly that these cells seem to congregate in the synovial fluid of patients with other forms of spondyloarthritis. Notice that in rheumatoid arthritis that is not the case. Now what's really intriguing is that these CD8-17 cells, a significant subpopulation are CD8 positive and are expressing the T-resident memory cell phenotype, both in the CD8 and the CD4 population. And particularly the CD8 cells are polyfunctional in their ability to produce a variety of cytokines, including interferon gamma, TNF, GM-CSF, and IL-21, in addition to expressing IL-17. So these cells have the potential to be major effectors in the skin and or the joint. So this was a very important study by Pencava in which they removed cells from the synovial fluid and the peripheral blood of patients with PSA, and they performed CyTOF, mass cytometry, to look at the different populations. And here you see memory CD4, memory CD8, naive CD8 cells, as well as monocyte macrophages, NK cells, and B cells from both the peripheral blood and the synovial fluid. When they broke this down into synovial fluid versus peripheral blood, they found there was a very marked expansion of the CD8 cells, particularly memory CD8 cells, to a lesser degree memory CD4 in the synovial fluid. They then went on to perform single-cell RNA-seq on these synovial fluid cells, and once again you see a number of cells that are expressing high CD8, transitional levels of CD8, and then this population here, which is a CD8 T-resident memory cell population, and indeed this was the population that was most expanded in the synovial fluid. You see T regulatory cells here, not so strong. There's some mate cells, and gamma delta T cells not really a major contribution to the synovial fluid in PSA. They then did T cell receptor sequencing of these CD4 and CD8 T cells, and here you see where they found most of the clones, which were in the CD8 and the T resident memory cell population. And then over here, they looked at those clones that were significantly expressed in the peripheral blood or in the synovial fluid. The red are those cells that are CD8. The triangles, the circles are the CD8. Triangles are CD4. So you can see most of these cells that are clonally expanded are in the CD8, and there's a high percentage of them in the synovial fluid, lesser so in the peripheral blood. So what about cytokine effects on tissue epithelial and stromal cells? Well, I'm not going to walk you through this whole paradigm of psoriasis. I'm sure you know it well, but the key effector cells are obviously T17 cells, which release IL-17 and IL-22, Th1 cells, gamma interferon, IL-12, and then you have this very important subset of T22, and these cells release IL-22. And it's well known in psoriasis as IL-22 is a major driver of keratinocyte proliferation. And these cells, keratinocytes, release a variety of cytokines and growth factors that stimulate the feed-forward inflammation that we see in psoriasis. So in psoriasis, IL-22 is a key cytokine that has very prominent effects. Interestingly, though, when you look at IL-22 in the synovium, it's elevated in the synovial tissues, and yet its effects are not nearly what we see in psoriasis. Indeed, if you knock out IL-22 in many mouse models of arthritis, you see no change in the phenotype. While it does have effects on cells in vitro, it can promote bone formation or bone resorption in assays, in vitro assays, and proliferation. And when in synergy with TNF, it can drive very high release of a variety of cytokines, IL-1, IL-8, etc. Yet in the synovial tissue itself, it does not appear to have a major effect, in direct contrast to what we see in the skin. So once again, tissue-specific events with the same cytokine. The other important area to mention are the stromal cells, which heretofore have not received enough attention. And this very highly quoted paper, beautifully done, Croft and others showed that there were distinct subsets of fibroblasts, and these were located in the subsynovium, and they were expressing Thi1, and they drove inflammation, whereas the Thi1 negative fibroblasts, which were located in the synovial lining, were releasing metalloproteinases and expressing rank ligand and driving joint damage. So here you have the important point that topography is essential to understand in order to gain insights into how the cells are interrelating with each other and carrying out various effector functions. Well, the other important point regarding spondyloarthropathy in stromal cells was published in a paper by Dominic Baton many years ago. And basically what he did was to take synovial tissue from patients with SPA in blue on the top there and patients with RA in orange-yellow and perform transcriptomic analysis focusing on muscle and myofibroblast genes. And what he showed clearly here was that the SPA tissues strongly express myofibroblast muscle genes in direct contrast to RA synovial tissue. So I think this is an area we should focus on more directly. We certainly have a variety of agents that target immune cells and immune cell cytokines, but this is an area that we may be able to gain even more handle on and improve our responses in spondyloarthritis. What about immune and resonant cell crosstalk and entheses in bone? Well, this is a functional model of enthesitis which really incorporates all of the features. The mechano sensation and immune activation with focusing on innate immune cells, likely acquired immunity is important here too, with the type 17 cells, Th17 and type 17, the innate immune inflammatory response, and then you have the mesenchymal proliferation, in part driven by the release of PGE2 and IL-23 and other cytokines, which then ultimately converge to result in bone remodeling through the activation of the cytokines and growth factors on monocytes to drive osteoclastogenesis and on osteoblast precursors to promote pathogenic bone formation. I'd like to talk now about mediators of osteoclastogenesis, which you know well, TNF, rank ligand, and IL-17, but it turns out these particular cytokines can have Janus-like properties. In other words, not only do they necessarily drive the resorption of bone, but they can actually also drive, in certain circumstances that we're just learning about, new bone formation. So here you have a paper from China where they looked at Wnt proteins in the spinal ligaments and sera of AS patients. And they found in this paper that a variety of Wnt proteins were elevated in both the tissues and the sera. They then went on to address the question as to whether low concentrations of tumor necrosis factor may be as important in driving osteoblast differentiation. And here you see that these are unstimulated, these are mesenchymal cells shown here, or osteoblast precursors, and they gave conditioned media and then increasing concentrations of TNF. And they were able to show, as reflected in the bar graph below, that the osteoblast formation was increased in doses of TNF between 1 and 10 nanograms per mil, but not at the higher dose. And so they propose and show the data in additional experiments in this paper that low concentrations of TNF can actually drive the differentiation of osteoblast progenitors. And over here, they look at Western blot analysis to show that in different, these are raw cells, which are an osteoclast precursor line, and monocytes, that when you treat them with no treatment versus TNF, again, these are low concentrations, that you can see an upregulation of the various Wnt molecules that they found in the sera and the spinal ligaments of these patients. So they hypothesized that low levels of TNF that you might see after you're treating patients with anti-TNF agents may be essential in driving continued new bone formation. In another paper, these authors showed that rank, which is released in vesicles, and the rank here is shown as sort of an antibody, that rank released in osteoclastic vesicles comes out and then can actually bind to rank ligand, which is on the surface of osteoblast precursors, and through reverse signaling in the mTOR pathway, upregulate RONX1 and drive bone formation. So once again, I'm coming back to that theme of complexity, plasticity, and the tissue microenvironment, which is driving pathologies which include synovitis, bone loss, new bone formation, enthesitis. And we have to understand what's going on at the tissue level with the specific cells at transcriptional understanding of how these events are unfolding in individual patients. So in conclusion, I've emphasized plasticity, which is so important at both the cellular and tissue level. I've discussed the important role of T-resident memory cells, which are known to be very important effectors in psoriasis. I didn't have time to talk about that, but that's been published by Rachel Clark and others, and in PSA. And there's also support now in RA and juvenile arthritis. The differential effect of cytokines on tissue epithelial and stromal populations with synergies and tissue-specific actions. And lastly, immune and resonant cell crosstalk, talking about the importance of biomechanical stress, TNF, rank ligand, and IL-17, which may have opposing properties in terms of their effect on bone, depending on the individual tissues and the microenvironment.
Thank you very much. Let's go to our post questions. Question number 6. Which statement describes best the role of IL-22 in psoriatic arthritis? IL-22 provides key proliferative signals to dermal fibroblasts in the skin. The major mediator of pathologic new bone formation. It is a major mediator of pathologic new bone formation in the spine. It is a critical activator of osteoclasts in peripheral joints. It mediates the recruitment of regulatory T cells to sites of inflammation, or it may play a limited role outside of the skin. 5 seconds to answer. 5, 4, 3, 2, 1. Okay. And the correct answer here is actually, if you go to the next slide, I'm a little confused because we have a
Got the wrong answer, wrong slide answer. There we go. So
yeah.
So this is an important question that Jürgen and I put together, because if you look at the rheumatologic literature, there are a lot of papers that describe a variety of in vitro effects of interleukin-22 on different cell populations. And as I mentioned in the talk, in vitro, this cytokine can promote the activation of osteoclast and the development of osteoblast precursors. It can drive the proliferation of different cell types. Yet when you look in the in vivo, its effects in the arthritis are actually quite limited. And I thought that stood in very strong contrast to what IL-22 can do in the psoriatic skin. So here, the real answer here is obviously its limited role outside of the skin. The first one was sort of a tricky question, provides key proliferative signals to dermofibroblasts in the skin. And as you know from Luis's beautiful talk, that is actually to the keratinocytes. So a bit of a trick one there.
Okay. Let's see. Can we locate the other question? Yes. So which of the following statements about tissue-resident memory T cells is correct? These are long-lived cells that continuously circulate through non-lymphoid tissues. They have a limited T cell receptor repertoire. They are lymphoid cells but do not have a T cell receptor. They express CD69 and CD103, or they provide a very rapid antigen non-specific cytokine response to local injury or infection. 5 seconds to answer. 5, 4, 3, 2, 1. Okay, I think we also showed in addition to the presentation, we also showed the slide already.
They're paying attention.
Yes.
Yeah, so they do express CD69 and 103, but I have to tell you there are a number of other markers now people have been identified for this cell population. I didn't get a chance to show it clearly, but in the Pankova paper, which they showed that ZNF683 is a marker of these cells. And there's also a paper in this online in ANR showing that this population is expanded in the skin of patients with PSA, psoriasis skin of PSA patients, but not in the psoriasis skin of psoriasis patients. Very interesting. And the marker they use there is a different one. So there's a number of markers that have been developed, but CD69 and 103 have sort of been the classic. These are a really interesting population, as I outlined, that may be critically important as polyfunctional cells in the psoriatic and likely the rheumatoid joint. The question that's been asked here is whether or not immune checkpoint inhibitors, such as those that inhibit PD-1, have been tested to block this population. Because as I mentioned, these cells often do express PD-1 and granzyme B. And I have not seen data on that yet, although I know people in their laboratories and murine models are looking at this. So I suspect that at least in preclinical models, we'll have an answer to that question pretty soon.
Yeah. And I think I can also highlight with regard to this question that these other options are not randomly chosen. So there's a whole bunch of innate-like lymphocytes that have been discovered recently and that might play a role in the spondyloarthritis family. So for instance, the mate cells, they have and iNKT cells, they have a limited T cell receptor repertoire, which would be the second option here. And then there is this family of ILCs, or innate lymphoid cells, which behave like lymphocytes and produce a lot of cytokines, but don't have a T cell receptor. So the third and fifth option here, they would be correct for these cells. But for the TRM cells, it's CD69 and 103. Last quick question before we go to the panel. Do you think that CD69 and CD103 might also be targets for therapeutic monoclonal antibodies?
Yes, and people are looking at that. Absolutely. The problem is that the CD69 is also a T cell activation marker, so it's not as specific as some of the more recent markers that have been identified. But people are definitely looking at targeting these cells in a variety of diseases.
Okay, thanks. Now I would like to bring in all the presenters for a final round of questions and answers. So unfortunately, we don't have a lot of time, but I think we can answer just a few more questions. One question here was about the role of TNF in the context of new bone formation and structural progression in axial disease, and some additional details on that.
So this is a paper that came out of China, very beautifully done, the one I presented by Lee and others. And again, they were able to show that in the tissues of MCERA patients with ankylosing spondylitis, that the Wnt pathway was very much activated, and that low levels of TNF can be a cytokine that can activate the Wnt pathway signaling and new bone formation. How that translates into axial spondyloarthritis and AS, I think, though, remains to be determined. They talk about the break hypothesis, where when you block TNF, there may be some bone formation, which is very controversial, and whether or not there's a subset of patients that have AxSpA where the TNF levels are actually very low and actually are promoting bone formation, I think is an open question. Kurt, you may have some thoughts on that, but I'm not sure of the direct translational impact here.
Yes, I do. It is like this, that TNF-alpha is an essential part of endochondral bone formation in the very early stage. So you need TNF-alpha to, in fact, to start this whole process of endochondral bone formation. So the whole discussion is now what happens in the spine. Is this membranous bone formation or endochondral bone formation? And I think to our, let's say, to what we have seen in our animal models, it's a bit of it's both parts. It's a bit mixed. You have components of membranous bone formation and you have components of endochondral bone formation. As a kind of case report, what we see is that we see some delayed fracture healing in patients who are under long-standing anti-TNF blockade. So in case of a fracture, and when the fracture is not very well positioned, we see somewhat more delayed union. So that might be also an indication that TNF-alpha is quite essential in the natural process of bone formation.
There was an earlier question, I want to bring this up again, about genetic differences between axial spondyloarthritis in AS on one side and axial PSA on the other. I find it very interesting, and Kurt, you commented on that in your presentation, about these differences also within psoriatic arthritis itself, in particular with regard to MHC association. Does that mean that we have to revisit the role of CD8 positive T cells in this group of diseases, or how do we interpret that?
I think I have no clear explanation yet for this. I think maybe Chris is better placed to comment on this, but it's for sure that, let's say, even the radiographic phenotypic characteristics in both diseases are different. You see less facet joint involvement in PSA compared to AS. You see more unilateral syndesmophytes or parasyndesmophytes, as you will call them, in psoriatic arthritis compared to ankylosing spondylitis, and you see more unilateral sacroiliitis in PSA than in AS.
Yeah, I completely agree with what you said, and Oliver Fitzgerald has shown us, and you presented some of these data, Kurt, about the differences in the MHC molecules in these phenotypes. On the other hand, there was a paper by Jaden and others where they compared axial PSA to axial SPA in a couple hundred patients, and there were a lot of similarities as well. So I think it's a very challenging area, and I'm going to be very interested to see when they start to focus the therapies of IL-23 inhibition in a very, very well-designed study, what outcomes we're going to see, and if it's going to be different between axial PSA and axial SPA. But there are clearly some different genetic underpinnings and some phenotypic differences as well. I agree.
I think it's partly deals with the fact, is it an enthesial disease or synovial disease? Yeah. And in the spine, there's only a part of the facet joint, the synovial, is covered by synovial tissue, because let's say the inner part is just the continuation of the yellow ligaments, and while the outside part is covered with synovial tissue. And I think there's a very interesting is an observation to see if this kind of particular therapies will have an influence. But after all, I think it's very difficult to find out if patients present with inflammatory back pain, is it just synovial driven or enthesial driven or a combination?
Yeah. Question to Luis. You talked about the central role of IL-22 in driving proliferation of keratinocytes in PSA. What about IL-22 as a therapeutic target in psoriasis?
It was attempted with fezakinumab, I seem to remember, and it did not lead to any results. Even in atopic dermatitis, where it might contribute to the acanthosis, the proliferation of the keratinocytes in the chronic forms of the disease, the results have been quite disappointing. Only those patients with high levels of IL-22 might respond a little bit. In my view, and most of my colleagues, IL-22 has a role, but it's ancillary, or at least it's redundant with the main drivers, which might be IL-17, but also IL-19, for instance, and some other cytokines, which, as a matter of fact, are produced by the keratinocytes themselves in a self loop. And it's quite interesting that the role of fibroblasts, or the effect on fibroblasts, has not been as much studied in psoriasis as it has been, of course, in the synovial tissue, but still there's a bit of fibrosis, and there are some changes which might be due or related to some cytokine interactions with these cells, but they don't seem to be so central as they might be in the synovium.
And there's a question in the chat to Niti, and that's about how patients respond to the large and increasing number of treatments that are available for psoriatic disease and spondyloarthritis. What are sources of information that are available for patients? Or you look at it from both sides as a physician and a patient. How should one approach this topic within the limited amount of time available in a clinic session?
Yeah, so I certainly advocate for patients to be their own advocates and to research information. However, there are, of course, sites and sources of information that may be more biased than others. At least from a psoriatic arthritis perspective, GRAPPA, the patient research partners, have put together lay versions of the treatment guidelines. Of course, the newest GRAPPA treatment guidelines are not yet available, but when they are, again, there's a plan for the patients to put together a lay publication related to that. In addition, the National Psoriasis Foundation, patient advocacy organizations in general tend to have better, less biased sources of information. I also find when I'm looking up things that the Mayo Clinic and Johns Hopkins also have very nice patient-oriented websites for educational purposes. Of course, my answers are a little bit U.S. focused because I am a U.S.-based physician, but I do know that many countries also have similar sources of information more pertinent to what's available in their country.
Okay, we have to wrap up. One final round, and maybe I can get a very brief response from all of you. where do you see the biggest need for making progress in understanding the pathogenesis of the diseases we've been discussing today?
Tissue-specific biology. It's going to vary from patient to patient.
I think the connection between skin and joint involvement, the evolution from one compartment or organ system to the other organ system.
My view, a contribution of local inflammation to systemic inflammation in psoriasis, and also the extension of the disease to the joints, and then regulation of T regulatory and tissue resident memory cells as a way to provide long-term remission. I would say these are the most important topics.
And for me, I would continue to advocate for better understanding in diverse populations if the data that we're talking about is applicable across the board or if there are differences.
All right. Thank you very much. Thank you very much to the speakers and panelists. I think we could have gone on for quite a bit longer. I would also like to thank Lisa from Spartan and Janine from Grappa for their hard work in getting this seminar organized. I would like to thank Pfizer for financial support, and I would like to thank Nucleus IMP for putting on the show in the virtual world. Lastly, I would like to alert you all to 4 more upcoming seminars in this series that will address other aspects of treatment and management of psoriasis, psoriatic arthritis, and axial spondyloarthritis. You can sign up for these seminars through the Spartan and Grappa websites, and the recording from the previous and today's seminars will be available through these websites as well. With that, I would conclude. Thank you for attending. And please let us know in your post-seminar evaluation how you liked the seminar and what we can do better in the future. Thank you very much and have a good day.